Hello students, welcome to EPG Pathshala. I am Dr. Shurmishtha De from Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today I am going to talk about surface plasmon resonance principle, methodology and application from paper techniques in Molecular Biophysics 1. Objective: surface plasmon resonance, that is SPR, is created at a thin metal surface by the coupling of incident light into the surface plasmon of the metal at an appropriate incident angle. The main objective of SPR are to quantify protein and other biological molecule at surfaces, to detect the interaction of drug protein, to detect the interaction of hormone protein, to detect the interaction of protein and protein, to detect the interaction of DNA and protein, to detect the interaction of carbohydrate and protein, to detect the interaction of lipid protein, and to interface with mass spectrometers provides discovery-based research in proteomic studies. Now, what is the principle of surface plasmon resonance? SPR is an optical phenomenon that provides a non-invasive, label-free means of observing binding interaction between an injected analyte and an immobilized biomolecules in real time. So, in SPR, no label, that is no tag, is required for the biomolecular interaction, like in ELISA and other method like Western blot, immunohistochemistry. In every method, it requires a tag at the label, but in this method, the tag is not required. It's a label-free method. So in this method, an optical detection process that occurs when a polarized light hits a prism covered by a thin gold metal layer. So over the surface of a glass, there is a layer of a gold metal. The free electrons at the surface of the biochips absorb the incident light photons and convert them into surface plasmon waves in between the two layers. It creates an electron density waves that propagate along the surface of the metal and create a very strong oscillating electric field near the surface, which is called evanescence wave. Total internal reflections strikes an electrically conducting gold layer at the surface between the media of different refractive index. So when the buffer or in a sample in a buffer is passed over the gold plate, the two surfaces form. One is the metal, that is the sensor surface, that is highly refractive index, and the buffer which passed over it is a low refractive index. So when the light falls on it, it passes from higher index to lower index and form a total internal reflection. The index of the refraction of a surface bound layer is proportional to the concentration of the bound molecule. So when the signals comes, it depends on how much molecules is binds over that ligand. It can determine the kinetics on and off, that is association between the two molecule and dissociation of the two molecule, that is off, for the interaction of a biomolecule with a ligand. Biacourt system. Biacourt system is used to utilize the natural phenomenon of surface plasmon resonance. Measurements with Biacourt system provide valuable information in a number of application areas, like to detect the concentration of proteins in body fluids, to screen the binding partners and ability for binding, for instance in drug discovery and biopharmaceutical development, to determine the interaction capabilities, for example epitope mapping with monoclonal antibodies, to measure the interaction kinetics and affinity made possible by real-time detection of interaction events. This picture is showing the Biacore machine, where this one is a sensor chips holder where the chips is placed over IFC, and this chips has a 4 flow cell where the molecules like protein, peptides or any biological molecules can be immobilized on the flow cells. This is West solvent bottle. After the reactions of this bimolecular interaction, the solvents from all the flow channels are collected here in this bottle. This is the running buffer pouch, which is contain a buffer to run the instrument, and this is an auto sampler needle which act as a robotic arm which collect the sample from the vials placed over the rack, and then it injected to the sensor chips through an injecting port over the IFC. These are the rack which contains 2 rack A and rack B, where the sample bottles are placed. This is pump 1 and this is pump 2. This pump 1 and pump 2 are used for circulating the buffer throughout the system. This picture is showing separately the pump. There are pumps which use for the circulation of the buffer throughout the system. So before starting the interaction, the prime systems is done to circulate the running buffer throughout the system, like auto sampler pump and connecting block, IFC chips, and then it finally comes to the West bottle. Here it's showing the needle, which are needle 1 and needle 2. Needle 1 is connected to pump 1 and needle 2 is connected to pump 2, which circulate the running buffer, and it also takes out the West solvent to the wash bottle, and this needle takes a burning buffer from this bottle and circulate throughout the systems in the machine. This picture is showing the integrated fluidic cartridge, which is the very important part in this Biacore machine. It's called IFC, consists of a series of channels in a hard polymer plate forming sample loops and flow channels for buffers and sample delivery. It contains 4 flow cells, flow cell 1, flow cell 2 and flow cell 3 and flow cell 4, which are connected to number of loops where all the samples and the running buffer comes from the bottle to the or from the sample bottle to the flow cells 1, 2 and 3, and after the interaction between the 2 molecules, it goes out through the other flow cells and collect in the West bottle. So this part is very sensitive, where the chips are placed over these cells and all the flow cells on the chips are corresponding to these flow cells of IFC. This is auto sample needle, a sample injecting needle which act as a robotic arm. It takes the sample from the sample bottle placed over the rack, and then it injects to this injector port, which is placed on the sensor chips, and the reactions takes place on the sensor chips, where already the another molecule is immobilized, and during the time of passing the analyte, again this injection needle takes a sample from the sample bottle and pass to the sensor chips through the injecting port. This picture is showing the rack where the sample bottles are placed, and these racks contain 6 rows, and each rows 10 sample bottle can be placed. So 160 bottle can be placed at a time over this rack. This picture is showing the sensor chips holder where the chips are placed on this holder and it pressed inside so that the chips goes and lie over that IFC. So this procedure is called the docking of the chips. Methodology which is followed in a Biacore machine for SPR technology. The required materials for start up the instruments buffer which are used: HBACP buffer, HBASP buffer, HBSN buffer depending on the reaction. Sensor chips are also used depending on which reaction will take place. These are the different sensor chips which are used, that is CM5, SA chips, nickel NTA, L1 and HPA. Amine coupling kit for amine coupling method, and the vials are used to hold the samples. Maintenance kits are used to maintain the instrument for washing and other method. Dissolved solution is used to clean up the machine. Bionormalizing solution is used to check the chips quality, and the computer is always attached to the system to run the machine, and all the programs are stored in the computer. The steps which are involved for the binding study of the molecules are immobilization, that is the immobilization of ligand on the sensor chips, binding of the ligand, regeneration, that is removal of the analytes from the ligand, and finally the evaluation. In evaluation, both the dissociation and association kinetics are measured between the two biological molecules. Immobilization on CM5 sensor chips by amine coupling method. The coupling reaction uses EDC, that is 1-ethyl 3,3-dimethyl aminopropyl carbodiamide, and NHS N-hydroxysuccinimide reagent. The protein ligand is immobilized by linking an amine functional group on the surface of the protein to one of the carboxylic group on the surface through an amide linkage. So after that, this ligand is immobilized over the sensor chips by amine coupling method. Ethanonamide is passed to remove the non-specific binding. Thiol coupling. Immobilization of ligands containing free thiols by the formation of disulfide bond with sulfur matrix on the sensor chips. So this is a sensor chip CM5 which is contain dextran matrix, EDC, that is 2,2-pyridinyl dithioethyl amine in 0.1 molar sodium borate, NHS N-hydroxysuccinimide react and activate the dextran surface of the chips, and then PDEA, that is 2,2-pyridinyl dithioethyl amine in 0.1 molar sodium borate react on it to activate the surface, and then the ligand containing SH groups are immobilized over it to form an SS bond with CONH group over the sensor chips. In aldehyde coupling method, 2 steps involved. Step 1, ligand is taken in the buffer, 100 millimolar sodium acetate at pH 5.5 is first oxidized by sodium metaperoxide. Incubate for 20 minutes on ice, then stop the reaction by desalting the mixture on desalting column eluted with buffer 10 millimolar sodium acetate at pH 4. In step 2, the surface is activated by injecting EDC and NHS in 1:1 ratio, and this activated surface hydrazine group is induced by injecting hydrazine or carbohydrazide, deactivate excess reactive group by injecting ethanol amine, immobilize the ligand by injecting the ligand solution, then stabilize the linkage by injecting cyanoborohydride. Now we are showing the immobilization on nickel NTA chips. So far we have discussed about the CM5 chips. Now it is a immobilization on nickel NTA chips. The surface consisting of carboxymethylated dextran matrix pre-immobilized with nitrilotriacetic acid. Chips activated by passing nickel sulfate solution on sensor chips. Nickel NTA is designed to bind histidine tag biomolecules for interaction analysis by metal chelation. So this is the profile is showing how nickel NTA chips is immobilized. So first the nickel chloride is passed, and then histidine protein is passed over it, and then EDTA is passed to remove the unreacted molecules, and then protein which contain his tags is immobilized over it. Immobilization on SA chips. The sensor chip SA dextran surface is pre-immobilized with streptavidin. So this sensor chip CM5 is used, which is immobilized with the streptavidin, that is in yellow color. The surface is washed with 3 consecutive injection of 1 molar NaCl in 50 millimolar NaOH, then biotinolated ligand. That ligand which is biotinolated is passed for short period over the streptavidin surface, which is in red color, to get bind with this streptavidin. Immobilization on SA chips. The sensor chip SA dextran surface is pre-immobilized with streptavidin. So this sensor chip CM5 is used, which is immobilized with the streptavidin, that is in yellow color. The surface is washed with 3 consecutive injection of 1 molar NaCl in 50 millimolar NaOH, then biotinolated ligand. That ligand which is biotinolated is passed for short period over the streptavidin surface, which is in red color, to get bind with this streptavidin. Now next is immobilization on L1 chips. Sensor chips L1 surface consists of a carboxymethylated dextrine matrix pre-immobilized with lipophilic group for rapid and reproducible capture of lipid membrane vesicle with and without membrane protein. The binding process involves diffusion of the vesicles to the surface and incorporation of the lipophilic structure on sensor chips L1 into the lipid membrane, non-covalently anchoring the vesicle. Immobilization on HPA chips. Surface over gold-plated layer is covered with long chain of alkane ethyl molecule to create a hydrophobic environment for adsorption of polar lipid monolayers. This picture is showing how the analytes binds over the ligand. So the ligands are first immobilized on the sensor chips, and then the analyte, this is the red one, it comes and binds over this ligand. After binding and the reaction is over, this analytes are regenerated and it goes regenerated and it goes out of the ligand. So this is the profile which showing that this is the dextran surface, and then where the one molecule, that is ligands, is immobilized and the analytes comes, and so association takes place, and after association the dissociation takes place, and then these analytes are removed by regeneration to get back the same surface where the only ligands are immobilized. That is the evaluation part by Biakor in SPR method. Kinetic study of biomolecular interaction. Measurement of association and dissociation constant by the interaction of analyte A and ligand B. A and B reacts to form a complex Ka, AB. Ka is the rate of association, Kd is the rate of dissociation. So Ka is equal to AB by A into B, Kd is equal to A into B by AB, where Ka is the association rate constant and Kd is the dissociation rate constant. At equilibrium of reversible reaction, the rate of forward and backward reaction become equal. Ka A into B is equal to Kd into AB. Equilibrium association constant is K capital A, which is equal to K small a by K small d, that is rate of association by rate of dissociation, is equal to AB by A into B. Equilibrium dissociation constant, that is K capital D, is equal to Kd small d by K small a, that is rate of dissociation by rate of association, which is equal to A into B by AB. The net rate of complex formation is given by dAB by dt, which is equal to K small a into B minus K small d AB. A is the analyte in solution which is constant C in the flow system. AB is the complex measured directly as R in RU. RU is the resonant unit which is obtained in the SPR system in Biakor machine. That is the signal which comes after binding. B is the ligand on the surface. That is, total concentration can be expressed in RU as maximum binding capacity R max, free concentration R max minus R. So dr by dt is equal to K small a into C into R max minus R minus Kd into R. At steady state, the net rate of complex formation is 0. So d capital R by dt is equal to K small a C into R max minus K small a into C plus Kd into R is equal to 0. So you can write Ka C R max minus R equivalent is equal to Kd into R equivalent. Kc R max minus Kc R equivalent is equal to Kd into R equivalent. So Kd R equivalent plus Kc into R equivalent is equal to Kc R max. So R equivalent multiplied by Kd plus Kac is equal to Ka into C into R max. 27. At equivalent state, R is equal to R equivalent. By rearranging the previous equation, it can be written as R equivalent multiplied by K small a by K small d into C plus 1 is equal to K small a by K small d C into R max. R equivalence multiplied by K capital A into C plus 1 is equal to Ka into C into R max. R equivalent is equal to K capital A C R max by K capital A C plus 1, where K capital A is equal to K small a by K small d, and K capital D is equal to 1 by K capital A. So K capital A can be determined from the plot made by R equivalent against C, and KD is the inverse of K capital A. The affinity constant is the ratio of forward and reverse race constant. K capital A is equal to K small a, that is on, by K small d, that is off. On is the association, off is the dissociation, and K capital D is equal to K small d, that is off, by K small a, that is on. This picture is showing the binding profile of ligand and the analyte, where in the equilibrium state, this is the equilibrium state when the analytes are binding at the initial stage, and then as it is increasing the concentration, then it's reached the equilibrium, R equivalent state. It is R equivalent state, and again after that it is two graph is showing the saturation step, which is called R max. This picture is showing how one molecule, that is antibody of SERTIN1, is immobilized on sensor chip CM5. First step is the pre-concentration to estimate the binding rate. In this step, the protein that is the here that antibody SERT1 is passed over the sensor chip to see whether it is suitable for binding on the sensor chips by pH and the concentration. So it's called pre-concentration to estimate the binding rate. And the second step is washing with ethanolamine. Then this antibody is removed by ethanolamine, and in the third step the activation of the surface takes place by EDC and NHS, where this mixture is passed to activate the surface by amine coupling, and then this is the different target to inject the ligand over the activated surface. 4 times it targets on the surface. Then after that the ethanolamine is passed to remove all unreactive group. This is another example which is showing how analytes bind over the ligand. In this Biacore SPR technology, the resonance unit increases with the increase of mass over it. So the RU increases with the increase of mass of the analyte over the ligand. So in the first graph is showing that the only 0.62 nanogram per microliter is passed when the resonance unit is 0. As the concentration increases to 3.12 nanogram per microliter, the resonance unit increases as the mass increases, and then another that is again from 3.12 to 6.25 nanogram per microliter, the resonance increases to 200, and when it becomes 18.75 nanogram, the resonance unit increases to 400, and then again another increase of a concentration, that is 26.62 nanogram per microliter, the resonance unit increases to 500, and after that finally the last concentration is 30.62 nanogram, where showing is near to 600 RU. So RU increases with the increase of mass of an analyte over the ligand. So student, let us now summarize what we have learned in the module. SPR, that is Surface Plasmon Resonance Technology, enables real-time detection and monitoring of biomolecular events and provides quantitative information on specificity. How specific is the binding between two molecules? If the molecule has not capability to bind with other molecules, it will not gives any signal. So how the specific is the molecule to binds over another? Concentration. How much of a given molecule is present and active? So how much concentration is required to bind between two molecules? Now, and then kinetics. What is the rate of association and dissociation? How much molecules will get associated with other one and how much will dissociate it, from which we can determine the rate of association and also the rate of dissociation. Affinity, how strong is the binding? This kinetic measurement will give us how the strong is the binding, what is the affinity of the two molecule. Then binding partner, provide the identification of binding target by linking SPR to mass spectrometry. So if the binding partner is not known, so it can be detect from the mass spectrometry, that is the proteomic detection. So from this technology we can detect the interaction of drug protein, hormone and protein, protein and protein, DNA protein, carbohydrate and protein, lipid and protein, and also the ability to interface with the mass spectrometer provides discovery-based research in proteomic studies.